Hi, everyone. Thank you for joining us today. My name is Nicole Suri, and on behalf of MEGIN, I'd like to welcome you all to this MEGIN Masterclass with our guest speaker, Sylvain Baillet. Just before we start, I'd like to ask attendees to please use the Q&A button on your screen to send in your questions at any time in the masterclass, and there will be time to answer questions at the end. This webinar will be available to watch on demand on the MEGIN website at a later date. So it gives me great pleasure to hand over to our guest who will officially kick off the webinar. Thank you.
Thank you so much, Nicole, for the kind introduction and for the opportunity for me to present our latest data and results to the MEGIN and MEG community. And thank you all for being here today. So indeed, I would like to update everyone on our current research on the neurophysiological basis of human perception and cognition. So this is a very ambitious program for sure. And I will try to explain how we try to narrow down those very broad questions to the mechanisms of neurophysiology, especially those that we think are at the basis of predictive processes that are so important in human perception and cognition. So I said these questions would be relatively broad and actually very broad. And indeed, I think we are all here today because we are somewhat interested in neuroscience. And that means we are interested in understanding the nature of human self. And with all the, with all these difficult questions, I mean, every time there is a, this kind of very broad interrogation, I think it's good to go a little bit outside our field and look at, for instance, what artists have to tell us, how they perceive basically these difficult questions. I'd like to cite a few quotes from Liu Fan, who's a Korean artist, painter, and plastician. And it's very interesting what he says, because he says that for him, nature is not an absolute truth. He has to constantly reconstruct it using perception through his physical sense. So it's an active process to perceive nature and perceive our environment. It's not a passive, purely passive operation, if you will. And he goes even further by saying that movement is very interesting, and the most fundamental movement of our human body, probably after cardiac muscle activity, is breathing. And basically, he has to feel that his breathing movements are somewhat aligned with nature's flux, whatever that means to him. And when he feels that this alignment is kind of broken, then he understands and he knows that it's time to end his day of work. So I think we should all do that, sitting at our desks, or scanning patients and participants when we feel that we are kind of misaligned with, our nature flux, and our body movements are kind of awkwardly misaligned with our environment. We should take a pause at least.
Sylvain, I'm just wondering if we can interrupt you. We're having some issues seeing your slides.
Okay.
We're just working to do so. Do you mind just emailing me the deck again, if possible? Oh, here we go. I can see it.
Go there.
Thank you. Let me try again.
Thank you. I can see them now, and everyone online, thanks for your patience as well. So back to you, Sylvain. Thanks again.
Okay, sorry about that. I'm not quite sure what happened. So this is Liu Fan. This is one of his paintings. And I hope you were able to hear what I was trying to say. So here you go. So if we want to understand basically the neural basis of perception and what makes us human, we have to interrogate also the nature, the biological nature of intelligence and reasoning beyond perception and actions. And of course, with all the the current affairs in AI, I think it's important also to understand what's the very nature of biological intelligence, is to maybe understand what distinguishes biological intelligence from AI. And we heard yesterday that GPT-4 was released by OpenAI. So one question would be, would ChatGPT pass the Turing test? And for that, I looked at a recent study published by two psychologists based at Stanford, and they reconsidered basically the Turing test, which has been, there's been discussions and some controversy about the validity of the Turing test. And they looked at the possibility to design a minimal Turing test to ascertain whether an agent is a human or a bot, basically that ChatGPT. And so what they did in their study a few years back is to ask 1,000 participants to basically come up with a single word that would convince a judge that this person is a human, or this agent is a human and not an AI bot. So they collected many responses from these 1,000 participants, and this is an example of the most frequent words that people came up with. And what's very interesting is that the largest semantic cluster are profanities that often refer to bodily functions. Actually, out of the most frequent words, about 200 of them belong to the same, which is about half, same largest semantic cluster related to profanities and bodily functions. So I thought that was interesting and also would make sense, right? That's bodily functions are what make us also, biological agents to begin with. So in a second study from the same paper, so they presented those words in pairs and they asked a human rater to designate the word that would be the most likely to be from a human. So they took among the 400 words that were, elected as to be the most humans from the first pool of participants. They then selected pairs and put them into in this form of competition. And this human rater would have to designate basically which word would be the most human-like. So I forgot about how many raters, but I think it was a good number, about 400, if I'm not mistaken. And at the end of the day, we had a winner, and the winner is poop. So indeed, not surprisingly, it's a bodily function or a product of bodily function. And so, and if you look at, the hierarchical, tree that puts poop at the top, if I may say so, in the Turing test. It's above love, it's about compassion, mercy, etc. So I thought that was very interesting because, again, this word refers to bodily function. It's eminently, not only related to bodily function, but it also evokes in humans an amused emotional response. So something that makes us or makes the value of this word eminently human. And I don't believe this is purely anecdotal, because if we look at the brain-body association, and that's basically what this experiment has been doing, I think, maybe we are looking at the essence of human cognition and the human self. So another technical word, if you will, to look at this is to consider the embodied self along, the history of the philosophy and neurophilosophy of cognition and consciousness. And it has been a long intellectual journey, very interesting to follow now, today in 2023. So we can look at Husserl and the phenomenon of experience. Merleau-Ponty, who has actually, really conceptualized the role or integrated role of the body with consciousness and thought, the process of thought. And Jonas, who's looked at the metabolism as a, a manifestation of the phenomenon of life. So, and designating in a relatively radical manner, sensing, acting, thinking as the only purpose of maintaining homeostasis for a biological agent like humans. And Francisco Varela, who has also revisited the phenomenology of a cell and embedded it, if you will, in modern neuroscience. So, and as we look at current AI, as impressive as it is essentially, for now, essentially disembodied. It struggles with taking action, and maybe that's for the better for now. But it has no notion of autonomy, no common sense, or very little. It's going to change for sure, but we are not there yet. and probably because it has no sense of maintaining its own homeostasis, its own integrity, which takes so much energy and time in us humans and most biological agents. So maybe we don't want to be there too soon, right? But this is definitely something to keep in mind. So now if we look at the brain in its environment, first of all, it's a brain and a body, and a body and a brain that interacts with multiple organs in that body. So it's very interesting to see recent, researches and studies, I should say, that looked at the interaction and actually how bodily functions like heartbeats influence sometimes very surprising, relatively higher order levels of cognition and perception, like interactions between the timing of heartbeats and the awareness in our visual perception. Gut functions also have been, hypothesized recently as playing extremely important roles in a set of neuropsychiatric and neurodegenerative disorders with the brain-gut axis hypothesis. Some also very exciting work by Catherine Talon-Baudry in Paris has looked at interactions between slow fluctuations of bodily functions in the gut with, again, perception, sensory perception, and cognition. So we can look at consciousness as one definition could be the embodied experience of oneself, and the body is a very crucial element of that. By definition of consciousness, it's definitely the envelope of our finitude and our interface to the world. And I think neuroscience has essentially been interested in looking in the top part of the body and of this figure. And now it's very exciting to see that there is a more holistic way to look at the whole body, including the brain, and the interactions between body and brain in human cognition, but also basic perception. So one theoretical framework that has been evolving over the years, just like the neurophenomenology of consciousness, as I mentioned before, is basically the notion that we construct the world based on not only the physical evidence from our senses, but also our representations and experience from interacting with similar situations as the one we are presently in now, based on, again, learning and development. So it has many names, it has had many names, and there's over the years, but we can look at early work by von Helmholtz about perceptual inferences, and basically perception as a probabilistic process. We can also look at the work of active inference, active processes, predictive coding, free energy principles that have gotten a lot of press over the recent years. But with important variations, we are essentially looking at the same thing, the same notion, that basically we construct perceptions based on physical evidence, but also a great deal of a priori information and mental representations of our environment. So I would like to make a simple proposition that it's basically paraphrasing what I just said, that complex behavior manifests via an active process and contextual brain-body interactions, so that the unified self can interact with its environment and take action, adapt its behavior to basically modify the environment, or actually just be safe, or actually benefit from the environment optimally, so to speak. But we have to resolve this as biological entities while facing a very important ecological challenge, which is that we need to adjust our behavior based on very noisy or at best incomplete information from our physical senses. So as I mentioned before, one possible solution would be to use our internal representations of the world that we have learned through experience, but also things that we can mentally construct and infer in a new situation to make causal predictions about what our actions, what the consequence of our actions would be in a certain context. So how do we do that? How does the human brain does that together with the body? So again, we're going to look through empirical evidence in the next few slides of the brain as a predictive embodied machine where basically the circuits, the neural circuits essentially driven by perception or involved in perception, are constantly interacting with higher order brain regions involved in motor actions, but also higher cognitive processes. And so the framework is that of hierarchical networks for predictive inference. And this is based, again, on a long tradition of work, but you can refer at some of these papers I've cited at the bottom that we've been, working on or producing in the lab over the past few years, and also with Stanislas Dehaene. So the idea is that when an external input reaches the brain through the senses, it's going to enter brain circuits through what we call usually primary input regions, so supposedly spatialized regions like primary auditory or visual cortex, for instance, and very rapidly will be propagated towards upstream into the hierarchy and reach directly or indirectly higher order brain network layers. And when you look at how the brain is wired, actually the vast majority of the fibers that are connected to primary input regions are fibers that re-enter those regions from higher order brain circuits. Like in the visual cortex, it's about 80% of the neural fibers and axons that actually enter the primary input regions from the inside of the brain, so to speak. So I'm sure many of you are familiar with the notions of bottom-up and top-down. And the proposition that we are looking at is that these top-down re-entries basically issue and convey the contextual predictions that are produced by these higher order network layers, so that basically what the primary input region is computed difference or an error, as some people call it, between, or a mismatch between what you've been predicting from a certain context in a certain situation at a certain moment in time and the actual physical input from the external world. And of course, depending on the amount of error, we're gonna be able to adapt our behavior through our motor systems usually, or make the appropriate decision to achieve our goal. But we're also gonna learn, because if the error is really detrimental to us achieving our goals, we need to adapt these internal representations so that next time the error is minimized. So this is a lot of, so to speak, conceptual black box hand waving, give it the name you prefer. So the challenge is to really look at the possible mechanisms that we can study, ideally non-invasively in the human brain, with MEG, for instance, as a tool. So the scientific approach to the embodied self, well, brain imaging is definitely one of them, and this is how we used to do it back in the 17th century. You've seen that image before, maybe. Today, with one brain, you can produce many, many maps based on the metabolism, the neuropharmacological processes or neurochemical processes of the brain, vascular aspects, electrophysiology, etc. And this is a shameless plug for the NeuroMaps toolbox developed in collaboration, or led, I should say, by Bradislav Mizic's team in collaboration with us and many others at McGill. So I'm personally very interested in looking at the brain in the real time of behavior and the real time of electrophysiological or neurophysiological process, because I want to constantly align basically observations of brain activity with observations of complex behavior, as be it perception or cognition. This is an image actually obtained with MEG, with source imaging, and we're looking at real-time brain activity over about 1 minute of a person's life, just resting state. So I think many of you are already aware of the huge complexity of what this data is telling us or trying to tell us. And for a long time, we've been considering, especially in the task-free resting state, that most of this activity would be noise. But there is greater evidence that actually this activity is eminently structured with the constant, basically, principles enacted of active information integration through these networks. So let me try to illustrate that with some empirical data now. So when you look at raw data, for those of you who sit at the MEG acquisition console, what you see is essentially those things. And this is just a subset of the many sensors available to us with this beautiful technology. So how do we make sense of that? How do we connect the dots between this messy complexity and the principles of active inference, for instance, in perception? So first of all, is it only noise? Going back to the previous question I was asking, when we look at all these, wiggly lines on the screen, is there any structure? Is it something that can be repeated, or is it a signature of a certain person's profile, just like the MRI structure of the brain is basically a picture of yourself, but a different one, only a different one. So we looked at this with Jason da Silva Castanheira in the lab, and what Jason did is it basically extend the notion of brain fingerprinting that had been developed in fMRI to neurophysiology and MEG in particular. And so the way it works is that basically you collect data from the same person over 2 different sessions. It could be on the same day, it could be sometimes years apart, as we showed in this paper. So you collect this data, these complex lines that don't make any sense. In our study, the average gap between the 2 sessions was about 200 days. And for that, we basically leveraged big data repositories like the Open MEG archive that we co-developed some years ago, and that is available and still growing as we speak. And we basically looked at very short recordings of task-free brain activity, only 2 minutes. And even we looked at the challenge of brain fingerprinting individuals with data durations as short as 30 seconds. So after that, Jason did some source modeling, so transforming the sensor data to brain images and extracted features, some of them, relatively basic, like power spectrum. So looking at the distribution of how much power brain activity produces in low versus high frequencies, but also connectivity. At the end of the day, the power spectrum are the most salient features, or simplest and most salient features at the end of the day. And then you can look at these different activities and profiles in all brain regions. You can vectorize basically these numbers. And these are the different participants, and each and every participant has a different signature, or as we call it, a brain fingerprint. And then you look at cross-correlation of the similarities between those different brain's fingerprints. And the question is, between day 1 and day 2, the question is, okay, are you more similar to yourself than anyone else in the cohort that you have tested? And if you are, that means that even several days, sometimes months apart, your brain activity remains a signature of yourself, just like your the tip of your fingers is a form of biometric measurements of who you are. So we are looking at inter-individual differentiation. Again, we are not identifying individuals. These people are, the data is anonymized. We're just looking at IDs that are encrypted, so P1, P2, for instance. We don't know who these persons are, but we might be able to tell, oh, this person, P1, is the P1 we saw on day 2. And we can look also at which brain regions play the most salient role in this identification. So first of all, we looked at an easy challenge of looking at back-to-back recordings and whether these recordings and brain fingerprints from MEG would be similar. And what Jason found is actually whether you look at connectome activity in red or spectral fingerprints in blue, basically you are very much able to differentiate between individuals above 90% accuracy from back-to-back recordings. And if you look at basically the different frequency bands from very slow to very fast, it's essentially the same story. You are very much able to differentiate between individuals. And the gray basically lines represent noise level, because one possible confounding factor could be that depending on when you scan the participants, the environmental noise would be different. and therefore would contribute to identification, but that's not the case by far. And then we looked at between sessions, on average 200 days between the sessions, the two different sessions from the same individual. And you see that, please mind the scale from 75% accuracy to 100%. You can see that we are above 90% accuracy. So this brain fingerprint, basically the complexity of brain activity, which is seemingly completely noisy and chaotic. Well, maybe it's chaotic, but it's certainly not noisy because it has sufficient structure so that even days apart, it remains somewhat stable. And it's actually remarkable that it's the case for pretty much all frequency bands. And even if you look at 30 seconds bouts of or segments of brain activity, you are still able to differentiate between people with this brain signature. So that's a remarkably stable property, if you will, of brain activity. And if you're interested in the stability and robustness of these estimators and of the different profiles, you can derive from resting state brain activity. I can direct you to Alex Wiseman, who is a postdoc in the lab, recent paper in Neuroimage. So then Jason looked at basically which brain regions play the most salient role in identification in which frequency band. So this is the intra-class correlation score. The higher the score, the most important this brain region is or this frequency band is for differentiating between individuals. And what you can see is that depending on the frequency band, some different regions are more important than others, more crucial than others. But what caught my eye is the importance of the beta band, which is actually producing the most consistently the greatest accuracy in differentiating between people from very short to very relatively longer time segments, and regardless of between versus within session identification challenges. And also see how basically every brain region seems to contribute to the identification or differentiation of the individual or the individualization of the fingerprint. And we have looked at the replication of that study in older adults and also patients with Parkinson's disease. And the good news is that there is also the possibility to differentiate between older individuals, 60 years old on average, and also patient participants. And of course, the interesting question is to use this brain fingerprint as a possible marker of the status of a certain patient, basically deciding about the the neurophysiological signature of a given syndrome or a given disease, and how a given intervention through drug or others can modify the disease trajectory and bring you closer to basically the fingerprint of a healthy person for your age group. So there is early evidence of that through a recent paper that Jason just put out on medRxiv, if you're interested. So now we looked at principles. Let's have a look at mechanisms. It's what we want to derive is a testable model for basically the instantiation of these predictive mechanisms in brain activity. So far, I've shown that resting state brain activity is essentially structured and is meaningful. Now we want to look into this spontaneous brain activity, whether there is any signature of predictive mechanisms. So for that, we need to do a little bit of modeling proposition. And what we are proposing is to move from this box-like representation of predictive inference into possible mechanisms that are testable, for instance, in MEG. So if we look at basically neural circuits, they are essentially tight interconnections between excitatory and inhibitory cells. And basically, when you look at how these neural circuits basically produce activity, even at rest, even in a dish, there is emergence of a spontaneous slow frequency rhythm in the human brain. Is essentially from delta to alpha frequency bands that basically reflect the net level of excitation of the circuit and of the cells. And intersped, modulated by these excitatory modulation cycles are basically bursts of high frequency activity from essentially gamma to high gamma. So this is a manifestation of phase amplitude coupling. I will come back to that later. And this is, we're going to assume that this organization is pretty, much true and instantiating everywhere in the brain. And these different circuits are, again, connected directly or indirectly. And when I say dynamical relaying, it's a certain function of networks to basically connect to each other through a relay. And we can think of the thalamus as an important relay for connections between two cortical areas. But it could be also a cortical hub, like some regions of the resting state networks, for instance. And what's missing from the picture are the beta oscillations. And what we assume is that the beta oscillations come in bursts. And these bursts are essentially driven by higher order neural and brain circuits to basically, again, interact with the and modulate basically the cycles of excitability of lower order circuits to basically align their timing essentially and predictions with respect to the expected inputs. So let me unpack that a little bit. So if we look at what's happening in this early, let's say, visual area, we're going to have this spontaneous oscillation around delta to alpha with a gamma bursts that are modulated in amplitude by the phase of the oscillation. So this is an extreme case of phase amplitude coupling, for instance, here, where you have gamma burst at the trough of the slow oscillation. And again, the red oscillation is a signature of the net inhibition or excitation of the circuits. And imagine there is an input occurring at a certain time and it's unpredicted at an unpredicted time. And what's going to happen is that there's going to be a phase resetting of the slow oscillation at a certain, caused by the unpredicted input because it was not aligned with the prediction from the higher order brain circuits. So that next time the input lies into the moment of greater excitation and lesser inhibition of the circuits so that it's registered properly by the brain networks. So there is a phase resetting so that, again, the timing of the endogenous oscillation, if you will, in the brain circuits is aligned with the expected timing of the external inputs, again, to optimize registration and eventually behavior. So we believe that this resetting is basically driven by the amount of error signal that is generated when the first input is presented, and that the phase resetting is driven by beta bursts, basically. And so that next time the beta bursts are initiated and sent to reset and or actually fix the phase at the moments when the, so that basically the oscillation or the rhythmicity of the red oscillation or brain activity in general in those early circuits is aligned with the timing of inputs. And we can go as far as basically looking at the possible role of these gamma oscillations, where basically they can also be adjusted of when they occur along the slow frequency cycle, so that basically they can receive, so to speak, inputs from the external world or also other brain networks at certain preferred moment of phase of the fast oscillation, because you see that the fast oscillation lower the level of inhibition even further below what is driven by the red curve here. So these are truly optimal moments when the circuits might be able to register external inputs. So I can cite the great work of Ole Jensen and others that have looked into this. I can also mention this paper that we have done with Therese Lennert in the lab in that realm, if you're interested to look into the details. So yes, phase amplitude coupling actually is something that is that you can easily model with computational, processes and models of networks between or circuits between inhibitory and excitatory cells. And indeed, when you do that, what you see is that, again, the cycles of greater excitation or inhibition are essentially driven by a rhythm, basically, a net rhythm that is emerging from the assembly. And that on top of this slow rhythm, you do have fast bursts of activity that is produced, but not randomly along the cycle. They tend to aggregate, as I mentioned before, in bursts at certain phases of the low frequency cycle. And this was demonstrated by Bob Knight and Ryan Canolti and colleagues with intracranial recordings in epileptic participants. And you do see that high frequency activity occurs, rhythmically and driven apparently by the slow frequency oscillation that is, dominant at that brain location. Here it's a prefrontal electrode, for instance. And you see these vertical stripes of greater and lesser high frequency power. So this is evidence of phase amplitude coupling. And what we did with Esther Florin, who was a postdoc in the lab and now a PI in Düsseldorf, is that we replicated this experiment with MEG, so non-invasively, in the resting state of many participants. And indeed, what we verified is the expression of phase amplitude coupling at different frequencies, low frequencies up to alpha, low beta. And you see again this replication of the vertical stripes of greater and lesser high frequency power driven by a low frequency phase. So again, from the apparent complexity of this brain activity that I was showing you before, we can extract some form of structure, and we propose that actually brain regions that express similar statistics of phase amplitude coupling, basically what it means is that they align their cycles of greater and lesser excitability and also express gamma activity in coherence between the different regions. And that could be a definition of a brain network, right? To have basically different brain regions aligning the timing of their processes in terms of excitability, in terms of gamma burst. And indeed, when we looked at resting state brain activity with Esther, we verified that statistically the regions that align their cycles of phase amplitude coupling basically can be expressed, can be decomposed in the typical resting state networks that we find in fMRI. And actually the fluctuations of these networks over time, they peak at a fraction of a hertz, which is very similar to the rhythmicity of what our colleagues in fMRI found. So it's interesting to see that there are oscillations in the brain, that these oscillations make us unique, that they are structured and interdependent. They don't live in different worlds, from very slow to very fast. But is there any association with behavior? Is it just anecdotal, or do they actually play a causal role? This has been a big debate in the community for many years, and I think MEG can provide evidence that there is, a causal role for these oscillations. So this is early work we did with Karim Gerbi, where we looked at basically the behavior, which is video game playing with a trackball. As you can see, it was a very simple game of maintaining the face of a cube, blue face in front of you using these trackball movements. And then we looked at the coherence of brain activity during the task with hand velocity. So behavior was hand velocity, and we looked at MEG brain activity. And the maps that you are seeing here are not brain activity maps in terms of amplitude. It's actually the greater, the brighter the color, the greater the coherence of brain activity with behavior, with hand movement. And not surprisingly, we found the contralateral sensory motor cortex and extended, regions like the SMA, but also the dorsal parietal cortex. And I don't know if you can see on your screens, but the ipsilateral cerebellum was also involved, as expected from hand movements. And when you look at this, you can find a coherence, a very clear coherence, for instance, in the M1 region between, again, the hand movement and brain activity. And if you extract a little, a little bit of this brain activity and the hand velocity signal in green, brain activity in blue, in contralateral motor cortex, and you filter around 4 Hz, which was the peak frequency in our data, you do find you can see with the naked eye that there is definitely very strong coherence. But the thing is that it's just an association. It doesn't mean that if you change, endogenously this frequency, that basically behavior would be suboptimal, for instance, or would change. So for that, you need to look at causal effects, and that's what we did with Philippe Albouy and Robert Zatorre in a series of recent papers. And we looked this time at working memory and auditory working memory. In particular, we presented short melodies of 3 notes to participants. We asked them to manipulate those notes, so either keep the same order or change the order in their head, in their mind, I should say. And then we presented them with another 3-note melody and asked them whether the tone or the pitch of the middle note has changed once you have, manipulated the sequence in your brain. So this is a relatively challenging, working memory task. But what Philippe found is that during this manipulation, there was a greater expression of theta oscillation, as you can see from this spectrogram. So time 0 is basically when you start manipulating the notes or the items in your working memory, and you keep them in working memory until you are probed with the second melody. And you can see indeed with the spectrum here, there is a strong peak around the theta band, and in a network that previous research has shown, including in fMRI, that these regions that this network was involved in working memory manipulation, for instance. But again, association, not causal role. However, what was interesting is that the greater the expression of these theta oscillations in those regions, the better the performance of the subjects. So what Philippe did was to manipulate the expression of these theta oscillations using TMS. So we brought back the participants and delivered 5 pulses of TMS, so 5 pulses in a second, around 5 Hertz then, which corresponded to the peak of the frequency we had observed spontaneously during the baseline test with MEG, and delivered these TMS pulses over the intraparietal sulcus, which was basically the region that expressed these theta oscillations the most, the greater, related to behavior. And the idea would be that we would boost basically those theta oscillations and hopefully boost behavioral performance if there were a causal association between theta oscillations and behavior, working memory behavior. So we did that with a simultaneous high density EEG, and you do see a boost of theta oscillations due to the TMS. And what Philippe found is that behaviorally there was a greater, greater performances following the TMS delivery at every trial. So not only did it boost the theta oscillation, but it boosted the performance of the participants as measured with D prime here with respect to baseline, no TMS, baseline MEG test, or arrhythmic TMS. So the same amount of energy delivered through TMS, but in an arrhythmic fashion. And the greater the boost in theta, the greater the boost in performance. So there was definitely a causal association between these brain oscillations and behavior. So for those of you who are not familiar with what you can do with these brain oscillations, especially in for instance in speech processing, but also in many aspects of perception. So you can basically track how these audio oscillations these brain oscillations, basically driven by stimulus, you can track them down into brain circuits. So for instance, if you present an audio stream, you can look at how the auditory cortex, but also to some extent, the rest of brain networks are trained by the external stimulus. So you can do that with frequency tagging, or you can do that by repeating basically speech phonemes, like in this paper with Emily Coffey and Robert Zatorre again, where Emily presented vowels repeatedly to participants. And if you do that a sufficient number of times, you can detect what specialists call the frequency following response, which is basically a response of the auditory system to the vowels presentation. And Emily showed that you are able to measure this frequency following response with MEG These are simultaneous recordings of EEG and MEG, and MEG is on the right. And very interestingly is that the notion was that this frequency following response would be essentially produced by the brainstem. So the auditory pathway in the brainstem with the cochlear nucleus, the inferior colliculus, etc. So very challenging for MEG to detect because it's so deep. But actually it's the brain tracking approach is so powerful that Emily was able to identify a cascade of onset responses from very deep regions very early on, like 6 milliseconds and above after stimulus onset, up to the cortical areas, auditory cortex, of course, much later, around 25-30 milliseconds downstream. So this is a plug for, MEG in general, I guess, but also the notion that you can really optimize your study design to boost your signal to noise and detect very deep responses, especially when they are very much tailored to boost the response from a specific circuit through brain oscillations. You can do that also with natural speech, and this is another collaboration with Robert Sartori and Sebastian Puschmann, where we presented speech bits and with target words, and it was a cocktail party design. And basically, you look at the response to the target words and to the target speech bits, ignoring you ask the participants to ignore the distractors input, sorry. And when you look at just a single, speaker situation with no cocktail party situation, you do find entrainment of a vast network of brain regions that encompass the auditory cortices, but also a greater language network, not surprisingly. What was in the cocktail party situation, you do find a similar, network. Of course, the signal to noise is a bit less because the challenge is greater, but you do find auditory cortices responding and also language networks. And what was very interesting to us was to find that the ignored speech part, or the speech to be ignored, was still encoded into the auditory cortex, but constrained to the auditory cortex. It did not penetrate, if you will, the language network, especially in participants who were very good at the task. So again, a very powerful, relatively simple approach of brain tracking stimulus inputs, but very powerful in testing sophisticated neuroscience questions, I think. So I will finish with this experiment that we did with Peter Darnhauser, who was a grad student in the lab, again, looking at interactions between or the predictive mechanisms in brain circuits. And the example is again speech. So imagine that we present a word in a speech. You listen to what I'm trying to say now. It's basically every word is in this context of the presentation I'm doing, right? And the presentation is even in a bigger context, and every word is in the context of a sentence, etc. So this context is actually, according to the predictive coding theory I was trying to explain, is actually essential in how this new word basically is processed. So there is you can basically qualify the context in terms of uncertainty, and physicists call it entropy of a context, depending on what I'm trying to say. Probably now the entropy is very high because you're wondering where I'm getting at. And then you can basically quantify entropy based on the probability of all the different words in your vocabulary in this given context. Okay, so this is a low entropy context because there are a few words with very high probability, and this is a high entropy context because pretty much every word is possible. And then you have surprise, which is brought to you when the word is actually uttered and presented to you in that context. So here this would be low surprise in low entropy context because the word that has been pronounced is actually the one that was the most probable. But this is not the case here. You experience a very high surprise. So you probably need to update your internal model of the context because you were a bit off. So essentially, if you want to test whether there are some processes in the brain that actually do that, these updates and assessment of uncertainty and updates of the models based on surprise, you will need to have access in real time to a certain measure of uncertainty and surprise. So if you were able to do that, then you would apply the previous methods I've shown briefly in the cocktail party situation, where you take each basically of these variables, apply a form of convolution like hemodynamic response function, but we call it temporal response function, that you can identify from the data, and basically try to predict the neural signals from the surprise and or uncertainty and other variables like the acoustic nature features of the speech. But the problem is that uncertainty and surprise are not observable, right? Because they are instantiation and manifestations of your internal representations of the context. So Peter came up with this idea to obtain some machine learned approximations of uncertainty and surprise by basically training a system to predict the next word. So please note this study was done only or published only 3 years ago. And that was before ChatGPT and the large language models, I guess, becoming so popular. And but still, the notion is the same. So actually, we use very rudimentary, architectures that are used in machine learning for predicting next words in a sentence. Definitely not with the sophistications and hundreds of millions of dollars OpenAI and others use to train their models, but still, with 30 tokens, the system was pretty good at predicting the next word or even the next phoneme. We trained it of 1,500 TED Talks, and basically we were able to have these predictions of the next word or even the next phoneme in a sentence. And once we had these predictions, we presented human subjects with a new TED Talk and also the artificial system with a new TED Talk that was not used for its training, and basically use uncertainty and surprise as predictors of neural activity. And what we found is that there were two principal components of brain activity related to the presentation of these TED Talk stories that were very much constrained to temporal regions. We had the primary auditory cortex regions with a temporal response function that looked very much like an event-related potential to words, as you can see. And we had a secondary region that was a bit more posterior and anterior, we call the association auditory cortex. And you see that it overlaps with the previous ones, but not entirely. But what does not overlap is the temporal dynamics of this brain region. And you can see it's much slower. And from the predictive, inference theory, we anticipate that these secondary regions would demonstrate a slower activity and more, responses to more surprising inputs, because they are basically, essential components for generating these internal representations of speech and speech content. And indeed, the cool thing is that what you can do is modulate, look at the differential response depending on the surprise and the context. Yes, so here you can look at the effect of greater uncertainty of the early visual response. So in a context where there is greater speech uncertainty with respect to the contents, you see a boost of early responses, which is very compatible with this, boosting of the input circuits, so that the registration of the input is probably more accurate, or the gain is actually greater in uncertain situation. It has no effect, this uncertainty, on the secondary order circuits. However, surprise has a very strong effect later in the response of the early circuit and of the secondary circuit. That is very much compatible with the idea that this conveys, the error signal, which is again the mismatch between what you were expecting and what you actually perceived, which happens actually with greater surprise. So what I'm going to do, I see that my time is running out, unfortunately. So I'm going to switch to my conclusion slides. And what I would like to share with you is a few takeaways. So that first, the brain is not a computer. it requires embodied cognition. I mean, basically manifestation of cognition is through the embodied self, and that homeostasis and metabolism certainly play a unique role in biological intelligence, and that predictive inference actually predicts as a theory that we are actively forecasting the context of our environment and trying to make sense of what's going to happen to us. If we predict well, we will perceive better and also have the most optimal behavior. And with multimodal human electrophysiology and MEG in particular, I think these are modalities that generate very complex signal, but that are the best for understanding complex behavior and symptoms in humans in the real time of behavior, in the real time of brain processes at the systems level, non-invasively. Of course, the complexity of the signals require that for interpretability, you have mechanistic models that help you forge hypotheses that you can test with the data. And that was the purpose of my journey with you today, to show how you can get there. And again, one of the manifestations of the brain as an embodied self, as an essential component of the embodied self, is that perception is an active process that is context dependent, and that you can test basically the different responses of the brain to the context and to the contents of the physical inputs, by the modulations of interdependent brain oscillations at different frequencies. So rather than looking at just one frequency, we have to look at a more holistic integrative view of these complex brain signals. One cool thing of MEG on top of all of the above, enabling all of the above, in my opinion, is that most of the methodology is transferable to all electrophysiology, including EEG and wearable devices, and that we can possibly also look at how this can transfer to impairments of perception and impairments of cognition and help patients eventually. So with that, I would like to stop here, thanking my lab and the students and staff who have helped me along the way. They are all doing the hard work, as you know. And I want to thank you for your kind attention. And thank you again, MEGIN, for hosting me today. And I would be happy to take questions if we have time.
Thank you. Yes, so thank you for your time as well. I just want to say, in the concluding remarks, it seemed that the slides disappeared, so we'll try and find a way to share this with the audience in the follow-up communication. So thank you for writing to me about this, and both Sylvain and I will try to coordinate how we can communicate just the last slide or two. Or Sylvain, maybe you can just go backwards one from this side, so I can see this slide now. Maybe then if we keep it on, everyone can see what the concluding
Yes, let me try this.
Thank you. Yes, awesome. Okay, I can see this. So yes,
Okay.
everybody, now you have the opportunity to see. So we really only have time for a question or two, and anybody feel free to write in something if you want to. But so can big data neuroscience help clarify further some of the mechanisms that you've mentioned?
So some of the data that I've shown are coming from large samples. So I mentioned the open MEG archives, but fortunately there are many others also resources available to everyone today. This is really a major leap forward in our community to see big data repositories available, often with really detailed phenotyping of the behavior at least and demographics of the participants. So there is the CAMCAN dataset, the human connectome, obviously, dataset is very rich and diverse in many ways. So I think for the kind of questions that we are looking at, especially related to resting state activity, brain fingerprinting, how it relates to demographics and maybe other types of complex traits of the person that can be documented with neuropsych testing, etc. I think these resources are extremely valuable because, again, we can not only detect smaller effects from bigger samples, but we can look at the greater diversity of the population and of the manifestations of these basically effects in a greater group of people. However, I still believe that, you know, very specific tailored studies, obviously with smaller samples because of the cost and logistics involved, are very valuable, and I've shown some examples, and the literature is filled with those examples. And I think if you really want to test specific hypotheses relating mechanisms to behavior, I mean, both approaches are essential, especially small sample approaches, hypothesis-driven testing. But again, it's a good time to do, you know, electrophysiology, MEG, and neuroscience today because of the open repositories that are available to everyone. Even when access to sophisticated devices like MEG is not given for everyone. So that's a fantastic way for everyone to contribute.
Thank you so much. So I think that's all we have time for today. Thank you for presenting this masterclass. It's been an excellent session, and to everyone that was attending, thank you for joining us today, and we hope to see you at future masterclasses. Thanks, everyone. Bye.
Thank you.